GTPase switch protein. And the activity of the GTPase is what? Its activity is the hydrolyzation of GTP into GDP and PI. Okay. So this acts as the switch protein and that alternates between an active state with bound GTP and obviously inactive state with bound GDP. Clear? So when GTP bound with the alpha, then it becomes inactive state, or it is in the form of inactive state. But when GTP is bound with the alpha, then it becomes active state. So when GDP is bound with alpha, it is inactive state, and when GTP is bound to alpha, that is the active state. So when G protein to an activated receptor, binding of G protein to an activated receptor, what is occurring here? That leads to the dissociation of GDP. When G protein that bound to the receptor, then the GDP that present in there, that is dissociated. And what is happening here? The G alpha is the G protein alpha, where the GDP has already bound, that is dissociated, and G alpha is bound with the GDP. Okay, and this when GDP concentration is very high, then G alpha is bound to the